Everybody, thank you so much for joining us. This is the November seminar on physical genomics. My name is Benjamin Keene. I'm here at CPGE, the Center for Physical Genomics and Engineering at Northwestern University. We're really excited to welcome a special guest this month, Professor Wendy Bickmore. Dr. Bickmore is Director of the Human Genetics Unit at University of Edinburgh. Just as way of background, I'm sure many of you are familiar with her. She received her undergraduate degree in biochemistry from University of Oxford before completing her PhD in molecular biology at University of Edinburgh. Following a postdoc in human genetics, she started her independent research group as a fellow of the Lister Institute for Preventive Medicine. Her current research explores how the non-coding genome regulates gene expression. Dr. Bickmore is a fellow of the Royal Society of Edinburgh, and of the Academy of Medical Sciences, and a member of the European Molecular Biology Organization, and an international member of the National Academy of Sciences. She was awarded a CBE for service to science and to women in science. Dr. Bickmore, thank you so much for taking the time to share your incredible work with us. Before we get started, one quick note for the audience. We'll do a Q&A at the conclusion of Dr. Bickmore's talk. So during the talk, anytime you have a question, feel free to type it into the Zoom chat, and we'll just address them at the end, or just wait till the end and you can ask then. If you're interested in the research and training programs here at Center for Physical Genomics and Engineering, you can visit us at physicalgenomics.northwestern.edu or check out our YouTube archive. We've got lots of interesting talks on there, and you can follow us on X, Blue Sky, and Instagram. Okay, Professor Bickmore, at this point, I can turn the Zoom over to you. Thank you once again for joining us today.
Thanks very much for that very kind introduction. I hope everyone can hear me okay. And thanks everyone for giving up your lunchtime to listen to me. And it's great to be able to share my enthusiasm for genome organization with those of you who are also interested in the way the genome is physically organized and how that relates to function. I'm going to talk to you today about my current research, which really focuses on how chromatin and genome organization relates to the function of genetic elements we call enhancers in the mammalian genome. And I want to do this from two very different ends of the scale in terms of chromatin structure. I'm going to talk first about some of our most recent work on the structure of the nucleosome as it relates to enhancer function. And then I'm going to zoom right out and look at kind of higher order 3D genome organization and enhancer function. So why am I interested in enhancers? Because we as complex organisms are not explained by the number of genes in our genome, which seems rather pathetic in comparison to other organisms that we think of as perhaps less complex than ourselves. But our complexity comes not from the number of genes in our genome, but the exquisite way that we can regulate their expression in time and space in different cells, in different tissues, at different times in development, so that we can reuse the same gene in very different biological contexts. We can take this gene here and activate it during early development of the eye to help shape the eye field. And then we can take the same gene and activate it in a specific set of neurons in response to a particular stimulation. Or the same gene could be reused and repurposed to help the heart deal with stress. So enhancers are fascinating genetic elements. They're embedded in the non-coding genome, and we still don't know how many there are in the human genome. Certainly hundreds of thousands, maybe millions or more. We don't know where they all are. We don't know which genes they regulate, and we don't know in which biological context most of them are activated. So there's a real knowledge gap there in human genetics. But we know that they're incredibly important because we know that a lot of the common genetic variation that we all share that changes our risk for developing a common disease, from cancer to neurodegeneration to diabetes, you name it. Those variants mapped by statistical genetics in the genome map for the most part not to the protein coding part of the genome, the only bit of the genome we really understand properly, but rather to the non-coding genome including in introns, but also in the intergenic space between genes, including signals that are hundreds of kilobases to millions of base pairs away from the genes. So genome-wide association studies have highlighted the importance of these elements, but I think it's been frustrating for the field that we haven't been able to really use this information, because it's very hard in these population-based assays to identify what the functional variants are and how they work and what genes they work on. But we also know that mutations, rare mutations that affect enhancers, also cause disease and cause very severe disease, often developmental disease. So here we know that single mutations in enhancers are the cause of disease through Mendelian genetics. And I'm just going to illustrate some of these to you here. So on the right hand side here we have a gene encoding an important transcription factor called SOX9. It's expressed in many different tissues. It's very important, for example, in aspects of development of the skeleton. So that, for example, if you have a deletion of one copy of SOX9, so you're haploinsufficient for this gene, the result is a very severe developmental disorder involving bowing of the long bones, problems with sex determination, and also breathing problems. However, mutations at this gene locus that don't involve the SOX9 gene itself but involve enhancers of SOX9 produce a subset of the phenotypes. So we know that the enhancers of SOX9 are spread over more than 1 million base pairs of the human genome. And so, for example, here I've shown you craniofacial enhancers for SOX9 that are 1.5 million base pairs upstream of the gene, and deletions of this enhancer or translocations that separate the enhancer from the target gene SOX9 result in the inability of SOX9 to be activated in a small population of cells called cranial neural crest cells, which are going to migrate up during embryonic development to form the structures of the lower jaw. So not having this enhancer prevents SOX9 being activated in those cells, resulting in underdevelopment of the jaw, resulting in problems, for example, with the tongue really blocking passage of the airway. So I think these kinds of examples, they're rare in human genetics, but they're incredibly informative in telling us how enhancers do and don't work. So, for example, they tell us that enhancers can be an extraordinary linear genomic distance away from the genes they regulate. I've given you the example from SOX9, which is perhaps one of the extreme examples. But we also know that single nucleotide changes in these elements can cause rare disease, because enhancers are the place in our genome where the tissue-specific transcription factors bind to regulate gene expression. So, for example, a single nucleotide change can ablate the binding site for an important transcription factor that's going to activate an enhancer. And here's an example here at the top of such a mutation in a forebrain enhancer, 500 kilobases upstream of an important gene called sonic hedgehog, which you're going to hear about later on. So that single nucleotide change stops that transcription factor binding in the right tissues, resulting in a failure to activate sonic hedgehog during brain development, with the result that there's a very severe developmental defect in the midline of the brain. But the molecular mechanisms can be more complicated. Here's an example here, which I've called autoregulatory. A single nucleotide change in enhancer that's 150 kilobases downstream of PAX6 actually alters the binding site of the product of the gene that it's regulating. And what's going on here is that during early eye development, PAX6 is being activated by a different enhancer, and then this enhancer under 50 kilobases downstream is supposed to take over through an autoregulatory feed-forward loop, but that fails in this case where there's a single nucleotide change, resulting in a very severe eye developmental defect called aniridia. And then here at the bottom, you can actually have gain-of-function mutations. Again, this is an example from sonic hedgehog, where it's best understood, where you have single nucleotide changes in the limb enhancer for sonic hedgehog, 1 million base pairs away from the sonic hedgehog gene. This enhancer is key to drive sonic hedgehog expression during a limb development, and single nucleotide changes that introduce one extra binding site for a key transcription factor of the X family results in an enhancer that's too good, that has too large a regulatory output, resulting in too much sonic hedgehog signaling in the limb, and then defects in digit development, for example, in digit number and identity. So enhancers are important, interesting, and we want to know how they work. So I'm going to start explaining the research by focusing in on the first level of chromatin structure, which is the nucleosome, of course. So in trying to find enhancers in the genome, people very often start by kind of a genome-wide approach, such as CHIP, trying to profile the genome for chromatin modifications that might be indicative of regulatory activity. And I think the most common one in the literature is acetylation of histone H3 on lysine 27. So this is a residue on the tail of histone H3 that's supposed to change chromatin function by acting as the binding site for effector proteins that will recognize that acetylated lysine. Very, very widely used, but actually really poor at predicting functional activity enhancers. So this is a paper we published a couple of years ago, a meta-analysis of all the data we could scrape on these different factors that are used to profile chromatin to look for regulatory activity. So transcription factor CHIP, DNase hypersensitive sites, presence of enhancer RNAs, hypersensitive site footprinting, K27 acetylation and attack sequencing, and then we correlated those data to the ability of these sites in the genome to drive regulatory activity based on different assays, such as the ability to bind transcription factors, to act in enhancer reporter assays, or to generate chromatin accessibility. And K27 acetylation was one of the poorest at predicting functional activity, largely because there's just a huge false positive rate. And moreover, in this really nice experiment from Christian Hellen's lab, they asked whether acetylation of K27 is actually important for regulation. And they undertook this heroic experiment where they actually mutated every single lysine 27 in all copies of H3 in the mouse genome. They showed that this completely obliterated, of course, K27 acetylation, but there was no impact on gene expression from active genes, even though the enhancers and the gene promoters themselves didn't have any K27 acetylation. Of course, things weren't normal in these cells because you need K27 to methylate it to silence genes properly. So without that residue, they found a defect in polycomb silencing. But in terms of activation, there was nothing to see here. So I think we often forget that actually the tails of histones are not the only place that you can get post-translational modifications. So we know, for example, that residues within the core of the nucleosome particle can also be modified. And these, of course, are not thought to act by acting as binding platforms for effector proteins. Rather, they're thought to act through directly changing the structure and the properties of the nucleosome itself. So we became interested in this several years ago in collaboration with Robert Schneider's lab in Munich, because Robert had been really looking at these core globular domain modifications for a long time. And we were able to show that this one here, for example, lysine 122 acetylation on histone H3, which is situated here close to the dyad axis of the nucleosome, produces a profile not dissimilar to K27 acetylation across the mouse ESL genome. We could find it at active gene promoters, but also at active enhancers. So this is actually the super enhancer of the pluripotency gene KLF4 here. So it's known in vitro that modification of lysine 122 affects the interaction between the histone octamer and the overlying DNA, particularly this dyad axis where there's only a single wrap of DNA going over the core particle. And we realised, looking at these structures, in fact, that there's another lysine residue, lysine 115, which is even closer to the dyad axis, directly making hydrogen bonds to the overlying DNA. So we wondered whether there was modifications of this lysine residue that might have interesting profiles across the mammalian genome. So we performed chip sequencing in mouse embryonic stem cells for lysine 115 acetylation, and it gave a very nice profile. Again, not dissimilar to K27 acetylation and 122 acetylation, but perhaps you can appreciate here that it appears to be a much sharper profile and with less peaks than the other two modifications. So this intrigued us, and we wanted to study further where we might be able to detect this modification. And so we'd also done 4SU sequencing, so we could get the nascent transcription rates at all the different sites in the genome. And here I've split the 4SU sequencing data into transcription start sites that are marked by CPG islands or that are non-CPG island promoters. And you can see both can be highly active here. If we look at K27 acetylation over these same sites, this is the typical profile of K27 acetylation around a transcription start site. You get these two peaks here around this dip, actually at position 0 in the transcription start site. You can see it's highest over CPG islands promoters, but you can still see an enrichment over non-CPG island promoters. In contrast, K115 acetylation was really different, and I'd never seen a profile like this looking at histone modifications in the genome. We could see, first of all, not this shape of two peaks in a dip, but a single peak right over the transcription start site. And almost all of the signal was coming from CPG island promoters, with very little coming from non-CPG island promoters. So if you want to find out more about this, this is just been posted as a reviewed preprint on eLife, and we're in the process of revising it. So if we look deeper into the CPG islands, which look really interesting, we then separated them out into levels of transcription based on the 4SU sequencing, expecting really to see the 115 acetylation really only associated with the highest transcribed CPG islands. But in fact, we found substantial levels of of K115 acetylation in all quartiles of expression. So substantial enrichment even over CPG island promoters that didn't seem to be active. So we wanted to explore this in more detail. So we took our ESLs and we differentiated them to neural progenitor cells, so a very simple differentiation system. And we separated genes that were upregulated, those that didn't change expression at all. And then we looked at whether the 115 acetylation changed at these sites or not. And we found a subset of regions in which transcription clearly went up as you went from an ESL to a neuroprogenitor cell. And as we might expect, these regions were marked by polycomb, the repressive mark in ESLs, which was lost as the genes became activated to become neuroprogenitor cells. But the K115 acetylation levels at these transcription start sites did not change. So the 115 acetylation was there before the genes became activated. So they were present at these CPG island promoters that are in the process of being repressed by polycomb, but are poised for later activation. So this was a pattern that hasn't been seen in the literature before, I don't think. So I've just got some examples to show you here on the right-hand side. So here's a gene, for example, FNF5. You can see from the 4SU sequencing, it's silent in ESLs, activated in neuroprogenitor cells. You can see that it's marked by polycomb in the ESLs, which is then lost in the neuroprogenitor cells. And I hope you can appreciate here that it's present, the K115 acetylation is present at this promoter even before it's being transcribed. So it's there before and it's there afterwards. And here's another example down here, this gene SLC22A3, not active in ESLs, activated in neuroprogenitor cells, loses polycomb during the differentiation, but K115 was there before the gene was activated. So a highly unusual histone acetylation mark that seems to be marking polycomb repressed but poised enhancers. So we'd also, as I pointed out to you, notice that with this interesting shape over the transcription start site of a single peak rather than two shoulders either side on the +1 and the -1 nucleosome. So this intrigued us because, of course, very often we think that the transcription start sites are, or they're often called nucleosome-free regions, or NFRs. But in fact, we know they're not completely nucleosome-free. So we done our chip on a native micrococcal nuclease digested chromatin, no cross-linking here. We sequenced the chip data in paired end mode, so we had the size of the fragments as well as whether they were marked by the modification or not. So we were able to plot our data in this way, in these plots that were first suggested by Steve Henikoff. So here what we're doing here's the transcription start sites piled up on top of each other. There's the 0 position. And on this axis here, on the y-axis, we've got the fragment size in base pairs. So this is canonical nucleosome size here. And then the intensity of the colour is the read depth. So what I'm showing you here is the input chromatin that we sequence. So you can see the -1 nucleosome, the +1 nucleosome. And here's the nucleosome-free region, but you can see there's signal in there. And it's on fragments, which I hope you can appreciate. They're just a little bit shorter than the standard nucleosome size. So the micrococcal nuclease is chewing into these particles more than the surrounding nucleosomes. If we look at the K27 acetylation chip, it's highly enriched on the +1 nucleosome, very little in this nucleosome-free region, what we should call probably a nucleosome-depleted region. If we look now at the 115 data, it's very different. And now you see this massive K115 acetylation signal over the 0 position, where these small nucleosome particles, and I hope you can see again this shift in the signal down to slightly shorter fragments. It's also enriched on the -1 nucleosome as well. So it looks like this modification is enriched on what we're calling a fragile nucleosome in the nucleosome-free or the nucleosome-depleted region of active transcription start sites. So to really show that these were nucleosome particles that were not canonical fully wrapped nucleosomes, we did sucrose gradient sedimentation to separate our micrococcal nuclease digested chromatin. So in these gradients, the particles were sediment based on mass and the frictional coefficient. So the fully folded octosome will sediment fastest, followed by the slightly unwrapped nucleosomes. So they have the full complement of histones within them, but the ends of the DNA are unwrapping. And then you've got the hexosomes, where you've started to lose subunits of the nucleosome, right down to hemisomes and smaller particles. So we took each of these fractions and then we sequenced what was in the fractions, so that's the input, and then we did chip on each of these fragments for K115 acetylation. So here's the input sequencing, again focused around transcription start sites. So I hope you can see this dip in the mononucleosome fraction at the position 0 in the nucleosome depleted region, and enrichment of particles sedimenting as fragile nucleosomes or hexosomes at position 0. And then if we look at the K115 chip data, we see almost no signal over fully wrapped mononucleosomes at the transcription start site, and a massive enrichment on particles that are sedimenting as fragile nucleosomes or as hexosomes, and even over particles that look like they might be hemisomes. So this modification is associated with nucleosomes with an unusual structure that suggests that they are very unstable. So we didn't just find K115 acetylation at promoters, we also found it at enhancers. And in contrast to CpG island promoters, where the modification was there before activation and after activation, the modification we saw at enhancers was very dynamic and correlated with activation. So I've shown you before these data for these two promoters here, where the 115 does not change as the genes become activated. But if you look at nearby ataxic peaks that are opening up as the genes become activated, you also see at these sites that they acquire K115 acetylation, or 115 acetylation is detected there. So we've got a dynamic marking at these regulatory elements. So if we can do the same thing and look at the size of these fragments, here I'm piling up all the data now around not transcription start sites, but enhancers in the mouse ESL genome that are bound by the pluripotency factor OCT4. So here you can see in the micrococcal nuclease sequencing this dip over the footprint of the transcription factors, so depletion of nucleosomes there. And this is the ataxic signal here, so here's the ataxic peak, and in fact the data is deep enough you can actually see the two footprints of the OCT4 within it. If you look at K27 acetylation, this region, remember this is very often used to profile active enhancers in the genome, you can see it depleted over the business part of the enhancer where the transcription factors are bound, whereas 115 is highly enriched over the binding sites of the transcription factors on both fully mononucleosome size fragments and also on subnucleosome size fragments. So in conclusion for this bit, I think this is a really interesting histone modification that's understudied and underappreciated, I think, and warrants further investigation. It seems to be associated with partially unwrapped nucleosomes at active regulatory sites, so it enhances and active promoters, but also at inactive promoters that are being repressed by polycombs, epigenetically ready to be activated at a later time in development. So as I said, it's very difficult in genome-wide association studies to find where the the interesting business part of the genetic variation is. K27 acetylation has not been very useful for doing this, and I think 115 acetylation might be a more useful mark. And some evidence that points to this, as it being having very high precision to identify functionally active regions, is this analysis that we did in the paper, where we took data from a star-seq experiment, which is an episomal enhancer assay done in mouse ES cells. So this is identifying sequences from the mouse genome that can drive regulatory activity on an episome. You know, it's an interesting assay, flawed of course because it's episomal. And here we've looked at that data and overlaid it with chip data for all these different histone modifications from mouse ES cells, and separated those two signals into those that correspond to sites in the genome that are attack positive, so they're associated with open chromatin and therefore likely to be functionally active, versus not open and accessible. So you can see all these other marks here that are commonly used, that yes, of course, they're able to identify attack-seq open regions of the genome, but that you've got a large kind of false positive region where you've got the star-seq is identifying sequences that are marked by these modifications, but actually those endogenous sequences in the mouse ESL genome look closed, not accessible. The 115 acetylation really stands out, I think, from all these other modifications in having a very small fraction of signals that are associated with closed or inactive chromatin. So we obviously need to look at many different cell types in many different situations to know whether this is really going to be a useful mark. So now I'm going to zoom out right to the other end of chromatin structure and look at three-dimensional folding of the genome in the nucleus. So I started off by talking about enhancers that are extraordinary genomic distance away from the genes that they regulate. So this is an area that really fascinates me as someone who likes to think about the genome at large scale. And I gave you examples of mutations, a particular regulatory loci that are worth exploring in more detail. And one of the classic regulatory regions where we know that there's long-range enhancers is the sonic hedgehog locus, and this is what I'm showing you here. So here's sonic hedgehog, here's a little gene over here, regulated by, we think, at least 20 different enhancers that act at different times and places in development and in the adult as well. And these enhancers are scattered over a large gene desert in our genome of almost 1 million base pairs in size. But some of these enhancers are also stuck in the introns of housekeeping genes, many hundreds of kilobases away from sonic hedgehog. And so in terms of thinking about how elements can work from such large genomic distances, and a massive leap forward in this field came from the realization that these large regulatory landscapes for genes like sonic hedgehog, or SOX9 indeed, align beautifully with the data from high C maps that identify these triangular structures that are called topologically associated domains. And I'm sure you're all familiar with looking at these kinds of data. So these data essentially are telling you that all the sequences within this triangle tend to, in quotation marks, interact, or at least ligate to each other more frequently than they do to sequences in the next door triangle, which might be closer in linear genomic space, but don't seem to be insulated from interacting with the sequences within the TAD. So that's a smoking gun, that there's something about this level of 3D genome folding that's important for how enhancers work, and perhaps particularly for how very long-range enhancers might work. So how are TADs made? They are made by this incredible process of cohesin-mediated loop extrusion. So this little movie over here is showing you the current model for how cohesin works. It's this SMC protein complex and ATPase that goes through this gymnastics where it reaches out, grabs pieces of the genome, and then bends over and kind of reels the DNA in, making a topologically entrapped loop that gets extended. So this is going on over our genome all over the place. Cohesin probably carries on for many tens of kilobases and then falls off the genome. Another molecule comes in and does the same thing. And in aggregate, that results in the cartoons we draw of what a TAD might look like. But I do want to emphasize that actually these structures don't really exist. They are the ensemble of data from millions of cells. So the fully formed TAD in which cohesins run all the way through until it's reached the TAD boundaries, which is where CTCF sites are bound, that lifestyle imaging tells us actually that's quite a rare event. So in most of the time, you've got these partially folded structures being formed, but nevertheless, in aggregate, they look like this. So can we do experiments that prove that cohesin-mediated loop extrusion and TAD formation are important for how enhancers work? That's actually been quite difficult to do. First of all, because cohesin is essential, you can't just do standard genetics and knock it out. Cells need it to divide. So we need to do something more clever than just do genetic deletions. And the enhancers, for example, of sonic hedgehog are not active in mouse embryonic stem cells and cells we want to study. So how can we study these enhancers in a simple system like an ES cell? So the way we've got around this is to, first of all, take control of cohesin acutely using degrons so that we can rapidly remove components of cohesin to the proteasome for degradation and study the effects at short term, not after cell division. And then we've studied the enhancers for sonic hedgehog, not by asking differentiation or development to activate these enhancers, but by designing synthetic transcription factors that will activate these enhancers ourselves so that we can deliver to these cells to turn the genes on. And we published this work several years ago now. And this has proved to be a very powerful approach, I think. So the first data I'm going to show you is using synthetic transcription factors that we built on the backbone of the TAL effectors. So these are DNA binding proteins from a plant pathogen where you can design a transcription factor that's going to recognize, for example, a unique 14-mer in the genome by changing amino acids at particular critical places within the DNA binding domain. And you can turn this into a transcription factor by fusing the TAL to a very strong transcriptional activator. And in this case, we've used 4 copies of VP16, this very strong virucidic activator. So the first experiment I'm going to show you is acute depletion of RAD21, a key component of cohesin. So we got mouse ES cells with an auxin degron tag on key cohesin from Rob Croce's lab at University of Oxford. This works beautifully. You add auxin to the ES cells, and within a couple of hours, the cohesin has disappeared and the TADs have disappeared. So this is the sonic hedgehog TAD. Now the triangle's turned around so you can see it as a square here. And you can see without cohesin running through the genome, the TAD essentially doesn't exist. You're left with basically a random walk polymer of chromatin. So that's great. We're able to really almost abolish completely this structure level of 3D genome organization. And now we challenge these cells with and without auxin, so with and without cohesin, with our synthetic transcription factors. And so first I want to stop and say, well, why did we do this? Because the literature said actually that cohesin is not very important for transcription. So when auxin-degrons for cohesin were first done, RNA-seq was done on these cells, and of course I assume that the authors were expecting to see, really quite dramatic changes in transcription, and the data were actually very disappointing. There were almost no changes in transcription, but this was steady-state messenger RNA, so these are genes that were already active and looking at the steady-state levels of RNA. That's very different from asking, can you activate a gene that's silent, has been silenced by polycomb, from an enhancer, so from the silent state to the active state, which is what we want to do. So our readout of activation of sonic hedgehog in this experiment is by counting the number of expressing alleles by RNA fish. So we're actually measuring individual alleles in the mouse ear cells. So minus auxin, so that's with cohesin and with the TAD intact, and then plus auxin without cohesin, no TAD there. So if we deliver our synthetic transcription factor directly to the promoter of sonic hedgehog, we can activate the gene nicely, and it really doesn't care whether there's cohesin there or not, no effect. So you don't need cohesin for transcription per se. We could also activate the gene from this enhancer here called SBE6. It's 100 kilobase away from sonic hedgehog. It's involved in activating sonic hedgehog in the neural tube. We could also activate synthetically from this enhancer, and we could still activate when there was no cohesin. Now we can also activate sonic hedgehog synthetically from these very long distance enhancers. This is the 460 kilobase brain enhancer I told you about that was mutated in holoprosencephaly, and here's the famous limb enhancer right at the other end of the TAD, the ZRS. So we can activate those synthetically with our transcription factors here, but only when there's cohesin running through the system and when the TAD is present. Once we degrade cohesin, we can no longer activate this sonic hedgehog from these long-range enhancers. So our conclusion that it was here that you need cohesin for the action of long-range but not short-range enhancers. So more recently, we've been following this up, looking at other components of cohesin. We've also adapted our system to work with dead Cas9 based transcription factors. So here we're using dead Cas9 fused to a very strong activator, in this case VPR, which is a bit stronger than VP64. Again, but we're activating from the same sites in the genome, and we're able to activate sonic hedgehog in this way. And in this particular experiment, we're reading out sonic hedgehog expression not by RNA fish, but actually using a fax-based assay, which I won't go into. We were able to show using this new system that we get exactly the same results when we degrade RAD21 as we did with the tal effectors. I'm not going to show you that data, but rather I'm going to show you some new data that's unpublished with this component of cohesin or accessory factor of cohesin called NITBL. So NITBL was originally identified as and suggested to be a loading factor of cohesin, and perhaps not actually involved in cohesin dynamics itself. But now the thinking is it's an active part of the loop extrusion machinery. We were able to degrade NITBL using an auxin, a DTAG this time, a different kind of degron. Again, the TAD melts away by high C. And now we challenge with our synthetic transcription factors at different positions at the promoter, the 100 kilobase. We also did 200 kilobase here, 400 kilobases, and 850 kilobases. No effect of depleting NITBL on activation from the promoter. So this is the log2 with and without the DTAG. No effect on that 100 kilobase enhancer again. So again, we don't need NITBL component cohesin to activate from enhancers. But as you get further and further away, you have more and more significant impact on the ability of long-range enhancers to activate their target gene, sonic hedgehog. So confirming the original result with cohesin. Why is this particular? And we could show that this was titratable. So if we used lower and lower doses of the DTAG, we could start to reduce NITBL dosage more gently. So we could bring it down to levels that were 70% or 50%, for example, and show that there was also a similar effect on activation. So the activation titrated very nicely with NITBL protein concentration as measured by FACS. Why is this interesting? Because haploinsufficiency, so lack of one functional copy of NITBL, it's associated with this very severe neurodevelopmental disorder called Cornelia de Lange syndrome. And in NIPBL, in Cornelia-de-Lange syndrome, although one copy of NIPBL is mutated such that it doesn't make a functional protein, in fact, there seems to be some compensation mechanism in patient cells such that the protein levels are only reduced by about 30%. But we can see here that even 30% reduction in NIPBL levels through this degron affects the activity of long-range enhancers to work. So our thinking is that Cornelia-de-Lange syndrome is probably a human genetic disorder in which there's impaired activation, particularly of long-range enhancers. And indeed, some of the phenotypes of children with Cornelia-de-Lange syndrome could be reconciled with, for example, failure of the sonic hedgehog limb enhancer. So we're getting these digit defects here. And also the long-range enhancers of SOX9. I hope you can appreciate the very small lower jaw of this traditional, this very classic facial appearance of Cornelia-de-Lange children. So I hope I've convinced you that cohesin is very important for how enhancers work in our genome, but particularly the enhancers that are a long genomic distance away from the genes that they regulate, certainly more than 100 kilobases or so. So how can we put this together in terms of how enhancers work? So some of the textbook models of how enhancers work I think are rather out of date. So these models where there might be just a very simple loop between an enhancer and promoter, we just don't see by imaging. Rather, we're thinking about enhancers as a point source of a signal that's short range and short-lived, so a diffusible signal. So we think an enhancer has a sphere of influence, and so therefore being close to an enhancer is important. It doesn't mean you have to touch it in physical space, but you've got to be within the sphere of influence. So in a cell where cohesin is running through, compacting the genome within TADs, the signal that's coming out of this enhancer is able to reach a gene that's many, many hundreds of kilobases away from it in the linear genome. And here's an example of imaging for the sonic hedgehog TAD, looking at the left-hand side of the TAD, the right-hand side of the TAD, and a point in the middle. And you can see that even this, though this is 1 million base pairs of the mouse genome, all of this DNA is in a very compact structure in the nucleus. When we degrade cohesin by adding auxin to the system, I showed you from the Hi-C data that the TAD melts away, but also just the physical distances in the nucleus between the enhancers and the target gene increases enormously. So now without cohesin, we have a situation where the enhancer is still sending out its signal. We think the sphere of influence is the same in terms of nanometers of radius, whatever it might be. But now the gene, if it's many hundreds of kilobases away from that enhancer, is now too far away in physical space in the nucleus to hear that signal from the enhancer, and therefore cannot be activated by the enhancer. For a more proximal enhancer that's 100 kilobases away from the target gene or less, it's sending out its signal, of course. And when you degrade cohesin, it's still doing that. And the chromatin overall decompacts, but because the gene is only 100 kilobases away, it's never going to get very far away in space from the enhancer, and therefore it's still able to be activated by this short-range signal that's coming out of the enhancer. I want to briefly, in the last few minutes, talk about another aspect of this level of 3D genome organization, and that's the edges of the TAD boundaries. So they have been suggested to help block regulatory activity, enhancer activation, from neighbouring TADs, so to stop enhancers over here, for example, activating sonic hedgehog. And indeed, we see no evidence of enhancer crosstalk across this TAD boundary in the mouse at all. Indeed, this is thought to be important in disease. There's several examples in the literature, really interesting examples, where the TAD boundaries have been compromised by DNA methylation, which prevents CTCF binding, and that has resulted in genes and oncogenes, for example, being activated inappropriately, producing tumorigenesis in very particular cell types. So, very interesting area. But I'm going to suggest that this might not be true across all TAD boundaries, because although we couldn't find any regulatory crosstalk across this end of the sonic hedgehog TAD, we could at this end here. So we were looking at sonic hedgehog expression here in mouse limb development. Here's the patch of sonic hedgehog in the developing mouse limb that's being driven by this limb enhancer here called the ZRS. And we happen to stain the mouse embryos for in situ hybridization for the RNA of this gene here called MNX1, which is only 150 kilobases away from the sonic hedgehog limb enhancer. The real target gene, sonic hedgehog's 850 kilobases away. And we could see, surprisingly, this shadow of expression in a pattern that looked identical to that of sonic hedgehog. So MNX1 stands for motor neuron homeobox. So this is a gene that's known to be expressed in the motor neurons and to be very important for patterning the interneurons of the developing neurotube. If you knock it out, you have very severe problems in neural tube development. Absolutely no defect in the limb whatsoever. We can't find any evidence in the literature that this gene has any business being expressed in the limb. So we think of this as bystander activation. And we were able to confirm our embryo in situ hybridization using RNA fish in tissue sections. We could find evidence of MNX1 expression alleles in the part of the limb bud where sonic hedgehog was being expressed. We didn't find any evidence of MNX1 expression in parts of the embryo where sonic hedgehog was being driven by proximal enhancers over here, over this end of the TAD. We could also, and we could show that the TAD boundary had an influence on this, because when we deleted that boundary, we made a 35 kilobase deletion here, we could show that this resulted in even more MNX1 expression in the developing limb. Didn't affect the expression of MNX1 in its real site of expression, the motor neurons. We could recapitulate this with our synthetic system here, so we could activate MNX1 in red here by our synthetic transcription factors that were directed towards the limb enhancer. And again, this was enhanced even further by deleting this TAD boundary. So this gives us first of all, it says that these TAD boundaries are not brick walls. You can get regulatory information passing over them. And it gives us an interesting scenario where we can address this model I've just presented to you, this hub-like model where the enhancer has a sphere of influences and is sending out signals, to the more classical enhancer-promoter looping model. Because in the looping model, one enhancer, yes, could activate two genes, but not at the same time, because the chromatin would loop over and one gene would come close and become activated. And then this conformation would have to flip around and the other gene come over for it to be activated. So you might expect to have activation of sonic hedgehog or MNX1, but not at the same time. Whereas in the hub model, there's no reason at all why you couldn't have both genes expressed at the same time. So this is what we tested in our system, and we're able to show indeed that we could find significantly more alleles where both MNX1 and sonic hedgehog nascent transcription was occurring on the same allele at the same time than you would expect by random. And when we went back and looked at the chromatin conformation by DNA fish of these alleles where MNX1 was being expression was being driven by the sonic hedgehog limb enhancer, we found very interestingly that this occurred in a situation where all the distances we measured by DNA fish were very small, i.e. when the whole chromatin region was highly compacted. So we've got regulatory activity going over a TAD boundary. Is this at all dependent on cohesin? So we played the same game with our synthetic transcription factors and degrading cohesin with an octindegron, and we found that yes, indeed it was. So when we degrade cohesin, we have less activation of MNX1 across the TAD boundary. So we should remember that when we look at high C maps and those triangles, these are not brick walls. You can find plenty of ligation products going between TADs, just not as many as you would expect. So this regulatory leakage across TAD boundaries is still cohesin dependent. So I hope I've convinced you in this second part of the talk that cohesin-mediated loop extrusion is key to enable enhancers in our genome to work over very large genomic distances. Our data overall are more consistent with a hub model that I proposed to you rather than a simple looping model of enhancer-mediated transcription. And enhancers can work across some TAD boundaries, but apparently not others. So it's clear we need to understand more about the different flavors of TAD boundaries that are in our genome, and that's something we would very much like to pursue. So I'm going to end there by thanking the people in the lab who've done the work I've talked to you about today. So the 115 work was driven by Yatendra in the lab and Elias. There's two postdocs in the lab. Here's Yatendra and Elias here. And the work on sonic hedgehog activation, the role of cohesin, was driven by Ian, Laura, Sheila, and also with assistance from Simon. And I'd also really like to thank the collaborators who've really helped us with both aspects of this work. And I'd like to thank you for listening. Thank you.
Thank you so much. That was awesome. So as I said, if you've got questions, I see we've got a couple in chat already. Jane, you had a question?
Yeah, great. Nice to see you again, Wendy.
Hello, Jane. Hi.
I was just intrigued by the H3K115 acetylation data and the fact that you show that it's present even when there's polycomb present. So I wondered, is this a bivalent issue, and how does it work?
That's a good question. So formally, we have not done. I think we'd have to do sequential chip for that to know whether it's on the same nucleosome. But if you look at the profiles, it's in the dip where there isn't any K4 or
Right.
any K27 methylation. So the K27 methylation and the K4 trimethylation are on the -1 and the +1 nucleosomes around the polycomb region.
Right, but maybe
It's just
they
not-
enable.
Yeah. So I'm sure it's related to the whole concept of bivalency of having this poised
Right.
protein state, but I don't think it's going to be exactly the same nucleosome particle. But
No,
formally,
I
we don't
wouldn't
know
think
that.
so either because of the dip, but I would just thought maybe they make it more permissive to have that
It could well be, yeah.
right.
So
But
we've got a circular question here. You know, does K115 acetylation makes the nucleosome more unwrapped, the region more accessible? But also, does it get acetylated because it's more accessible?
right, exactly, yeah. But you
And
never
that's
see it without the, you never see it where you suddenly get activation and it's not there before the activation. So
we haven't seen an example of that. But also, we don't have very good temporal resolution of this differentiation.
Right.
It's days later. I think we need a nicer acute system than we've done so far.
Right. Okay, that's great. Thanks very much.
Emily, did you want to
Yeah. Hi, Wendy.
Hello.
Just nice. Let me see if I can turn my camera on. Nice to see you. So just hi, and following Jane's question, and sort of you alluded to it, my mind was sort of thinking that maybe the unwrapping is happening and allowing the acetylation, and then that sort of made me think, is there a specific hat that is sort of sensing this unwrapped nucleosome, and it wouldn't be a general histone acetyltransferase, but something more specific. So I wonder if you have any ideas.
I mean, formally that could be the case, but inhibitor experiments that I'm not quite happy with, but certainly strongly suggest that it's P300. And I think this is the trouble with the hats. You know, apart from MOF, which is very different. All this class of acetyltransferases has got really broad substrate specificity. They've got a very shallow substrate pocket. So I think they'll just acetylate any lysine that they get a sniff of.
Exposed, yeah.
Yeah. So I think it's P300. I'm sure that there's a very significant role for remodelers acting at these nucleosome particles in helping to allow the acetyltransferase to get in there. And then perhaps the modification, helping to further destabilize the nucleosome. So I do think it's kind of a chicken and egg circle we've got going on here.
Right. Yeah, it's very exciting.
I think it's going to be a great tool to add to the toolkit for profiling chromatin.
Rivan, do you want to go?
Yeah. Hi, thank you so much for this talk, Dr. Bickmore. I'm a graduate student in Dr. Bachman and Dr. Schleifer's labs, and the work you showed here was very elucidating. My question is on your work with high C and trying to assess the degree to which there would be crosstalk between enhancers. You had lots of interesting models on the distance at which there wouldn't be crosstalk or the enhancers would have an effect. And then also when there are, like, I guess, some open questions on whether or not TAD boundaries have an effect on whether there would be crosstalk. Just given that high C data is on population data and there is heterogeneity among individual chromatin conformations, would you have interest in imaging specific enhancers at the single cell level to try and see if that can help shed light?
Absolutely, and exactly why I kind of advocate doing things like high C and imaging at the same time, where it's because you get very different information. You know, on one hand, you're getting this kind of population aggregated, very deep, very interesting sequencing based data, and on the other hand, you're getting this kind of single allele imaging data. But if you think about the high C first, you know, those boundaries, you know, you can measure the insulation score of those boundaries. And really, you know, the read depth there in high C only goes down twofold. There's loads of ligation fragments going on across TAD boundaries. So what we think of as boundaries is probably a bad word for them. I can't think of a better. They're very porous boundaries for cohesin, let alone anything else. So viewing it from that way, actually, it surprises me that actually there are seem to be some TAD boundaries that absolutely do stop regulatory information going across them, as shown in some of those cancer studies there and some of our own work. So I think we just need to know more. Maybe there's more going on at TAD boundaries than we think of. It's not just CTCF interacting with cohesin. There may be other factors at play there that we don't understand yet. So I think, you know, I've shown you a few, you know, one example basically where it doesn't seem to be a very simple relationship between a boundary and no crosstalk. I'm sure there's going to be all kind of different flavors of TAD boundaries that have different insulation effects in terms of regulation, and we just need to look harder.
Thank you. This was so fascinating.
Thanks, Ravon. There's another question in the chat. I don't know, Wendy, if you just want to.
Yeah, of course, Lara. Yeah. You might have mentioned it, and I didn't actually, and I might have missed it. You didn't. What's the relationship between chromatin accessibility, I assume as assayed by attack sequencing, and chromatin looping? Is there a direct and clear causal relationship, and what comes first? Actually, I don't think. Oh, hello, there you are. I don't think there's any relationship at all. Actually, chromatin accessibility, as measured by, you know, attack or MNAs, is local, and it's basically telling you that something else is bound there, usually a transcription factor, that stops a fully formed nucleosome being there. So it's a highly local structure. Whereas chromatin looping in terms of enhancers and promoters, even though I don't think they directly contact each other, is about proximity in nuclear space, I think, and has no relationship to the actual what's going on at the nucleosome level.
So you don't expect that modifying chromatin accessibility, for example, making chromatin more accessible, would result in changes in
I don't think
because
so, no.
chromatin is more accessible to cohesin itself, for example.
No, the only link may be to where cohesin loads. So there's some really interesting data from both high C and micro C that suggests that promoters and enhancers, which of course are accessible, form these what are called stripes or flares in high C data, which suggests that actually cohesin is being loaded there. So cohesin loop extrusion may start at accessible regions. But then the overall 3D structure is not related necessarily to that local site. So you're right, in a way there is a link, but it's not a direct one, it's an indirect one. So I think this idea of where cohesin's loaded and which direction it's going in is very interesting, but not really.
Can I jump in and make a comment? Because
yeah,
we showed that there's a very strong relationship between accessibility and the signal strength of CTCF, related also to the signal strength of cohesin. So at CTCF sites, if you have an accessible region, the CTCF signal is stronger in general. It's not the only thing that you can have them at inaccessible sites as well, but the cohesin signal tends to be weaker. So
but I guess there it's kind of confounded by this actual molecular interaction between CTCF and cohesin as well.
Exactly.
And would you expect, so yes, so the modifying chromatin accessibility would change cohesin loading and maybe the
Yes,
like
yeah.
the what about the other way around? So would you expect that knocking down, for example, cohesin, or the other way around, pushing cohesin on the chromatin would change chromatin accessibility, in which way?
Ooh, I wouldn't expect it would, but I'm not sure if anybody's looked. I can't think in the literature. We haven't looked. Maybe we should. It's an interesting experiment. I don't think it would, because cohesin is moving incredibly. It's not staying there. It's moving very fast. It goes 10 times faster than polymerase. So I don't think it's going to kind of leave a footprint behind of accessibility, but I don't think it's formally been tested.
Yeah,
Let's go
I
back.
have this.
Yeah.
Yeah.
You need to go back and look at the literature and look at that.
Thanks.
It's good suggestion. I think the fact that in the first paper where they did that degradation of cohesin and just did RNA sequencing, the fact that transcription didn't change suggests that there wasn't really any change in chromatin accessibility.
Thanks.
Okay, Bryce, do you want to go?
Thank you so much for your talk, Dr. Bickmore. I'm very curious about your sort of zone of influence hypothesis with the enhancers, because it's rather different from the canonical mediator, modulated, or conferred effect of them. Do you have any ideas or hypotheses about what that sort of second messenger or diffused signal might actually be?
Yeah, good question. So in fact, I didn't come up with this idea. I kind of jumped on it based on it was a really interesting opinion piece authored by Xavier Darzac and Bob Tijan in Berkeley several years ago. It was in Genes and Development. And I thought, oh, this really makes a lot of sense. And they kind of talked through different models of enhancers and promoters and whether the data were consistent with looping or not looping. And they concluded that the data in the literature was not consistent with simple enhancer promoting looping, but rather they suggested this diffusion-based model. And there, when I was originally thinking about these ideas, I thought, well, maybe it's just a transcription factor that's diffusing, or a mediator or some other protein component. But they argued that no, it's not as simple as that, because that wouldn't give you the specificity you need or the short range that you would need. So they suggested in that piece, and they called the piece is called the transcription factor activity gradient. They suggested that the signal, the second messenger, if you like, is a post-translationally modified protein, which has got a short half-life because the modification's getting taken off. And what would that modification be? Well, we don't know, but a good candidate would be acetylation, because P300 acetylates anything, any protein nearby, it will acetylate. And of course, the nucleus is full of histone deacetylase, is taking that modification off. So there, it kind of smells plausible that it might be an acetylated protein. It might be a component of one of the coactivators, it might be polymerase itself, who knows? But that's what I'm thinking about at the moment. It might be a post-translation modified protein. I don't know how to test it, but thinking about it.